So our fourth learning unit is on electron transport and oxidative phosphorylation. And so, we've been working with this equation since the start of biochemistry, and we finally now in this last learning unit are going to put all of the detailed pieces together. So again, fuel becoming oxidized, losing its energy as electrons, which are transferred to these middlemen, NAD plus and FAD. Their reduction into NADH and FADH2 is a way to harvest and save those electrons. Again, now we're going to talk about how we save the energy of those electrons. As these species are oxidized, their electrons again go to the electron transport chain. So during the electron transport chain, we're generating a proton gradient, and that proton gradient is dissipated in the process of oxidative phosphorylation to generate most of the ATP that we have from fuel metabolism. As the electrons finish their route through the electron transport chain, the final electron acceptor is oxygen. So oxygen in gaining those electrons is reduced to water. And fuel in its full oxidation, carbon in its fully oxidized form, is carbon dioxide. So again, we are going to, in the rest of the course here, now expand our understanding of fuel metabolism to not just be carbohydrates, which we've dealt with primarily thus far in glucose through glycolysis, and then pyruvate being derived into the citric moved into the citric acid cycle. So just kind of a little reminder here that we've mainly been focused on carbon, hydrogen, and oxygen when we're talking about carbohydrates. But we're going to move after a chapter on photosynthesis into metabolism of fats. So we'll talk about fatty acids, which are primarily just carbon and hydrogen. We've got a more oxidized fuel in carbohydrates, which is again why it's not worth as much as fats, because it's already further along that fuel oxidation continuum. And then after we do lipids, we'll talk about proteins. And the unique thing with proteins is they have the element nitrogen. So we'll talk about how we deal with nitrogen. Sort of a main big picture thing to think about here is amino acids basically have an alpha keto acid skeleton part and then that nitrogen atom. And so the first thing that we'll talk about in our amino acid chapter is how we deal with the elimination of nitrogen. And your body does that largely through metabolic processes in the urea cycle. And then once you have this alpha keto acid skeleton, that's essentially a carbohydrate. So we find some flavor of carbohydrate in the metabolic processes that we've discussed so far that we can kind of play into that. So that was a very quick, but again, overview of what started out as a very simple equation for us at the beginning of Biochem 1. And as we've mapped on and sort of understood these different pieces, we're now in this final learning unit to understand the details of those pieces. So again, highlighting where we get those electrons, we indicated very generally where they were here, but reminding us that whether it's carbohydrates or fatty acids or amino acids, at the end of the day, they're ultimately funneled to acetyl-CoA. So just a little bit of a review in terms of things that are important to remember as we continue through these metabolic pathways chapters. Even though we might not be studying all of the details of glycolysis, there are some important pieces that we do want to make sure we remember. Importantly, that we have cytosolic NADH that's generated there. We also have cytosolic ATP that's generated. Again, just a little bit in that glycolytic pathway. Endpoint of glycolysis is again pyruvate. Pyruvate undergoes further oxidative decarboxylation by pyruvate dehydrogenase to generate acetyl-CoA. So that process is mitochondrial, so we're generating some mitochondrial NADH. We haven't talked about these processes yet, but we talked about them very generally at the end of Biochem 1. Fatty acids are metabolized to acetyl-CoA in a process called beta oxidation. That generates both NADH and FADH2 in the mitochondria. So we're going to have mitochondrial forms of those two reduced cofactors. And again, we talked very superficially on the last slide here about we have amino acids that we deaminate via the urea cycle, and the alpha keto acid skeletons are either of a glucogenic or a ketogenic form, depending on where they're able to feed back into metabolic pathways. But ketogenic forms specifically can be funneled to acetyl-CoA. And then once we're at acetyl-CoA, what happens with the acetyl-CoA basically has to do with whether we're in a fed state or a need state. And again, global or a local need state means that acetyl-CoA is going to be further metabolized in the citric acid cycle to generate mitochondrial NADH and FADH2. Again, those, as we're going to see in this chapter, are converted into a proton gradient via the electron transport chain, and ultimately that proton gradient is dissipated to generate the ATP that's needed locally. Again, if we are in an ATP or a fed state where we've got excess acetyl-CoA, and there's only some cells that are able to do this, the liver is one of them, but fatty acid synthesis can occur. We'll learn about that process in a couple of chapters, but we're basically going to synthesize fatty acids from the 2-carbon acetyl-CoA units. We're going to then assemble those into triglycerides, and then we're going to store those excess fats, again, if we're in a fed state. So just highlighting here are, again, the 2 processes of glycolysis and then the citric acid cycle, highlighting here all the different spots where we do make some NADH or FADH2. And again, noting that we do have a little bit of cytosolic NADH that we're going to have to deal with because electron transport occurs in the mitochondria. So all these flavors happen to be produced in a cellular locale where they're going to be immediately used in the electron transport chain. So we spent a little bit of time at the beginning of this learning unit as well talking about this very important organelle called the mitochondrion. And what's unique about the mitochondria as an organelle, similarly as we'll see in next learning units, the next learning unit on photosynthesis, is this is a very unique organelle. It's not just a separate compartment where we're able to do unique biochemistry. It's actually an ancient bacteria, or at least that's what the endosymbiotic theory tells us. And there's a lot of evidence for that, especially the fact that there's separate DNA that's found in mitochondria that's unique to mitochondria, and as we'll see in our next learning unit, unique to chloroplasts. So we've got these little ancient bacteria that are living within our cells. What's kind of interesting to think about some just facts about them when we talked about their story. Right now, there's only 37 genes that are kind of stored in mitochondrial DNA. They code for 22 transfer RNAs, 2 ribosomal RNAs, and 13 of the many proteins that are found in the electron transport chain. But originally, it's thought there were about 1,000 genes that were in mitochondrial DNA. There still are about 1,000 different genes that are used to make proteins that are found and function within the mitochondria, but they've been translated and transported over into the nuclear genome. And so the thought is that there's this very sort of minimalistic essential set that's retained within the mitochondria's own DNA, but they've kind of farmed out all of the other genomic material to our own nuclear DNA. Kind of something else that was unique to think about when we were talking about the story of mitochondria is thinking about how many mitochondria are found within cells. And so, for example, something like red blood cells we learned about do not contain mitochondria. And that's important because remember when we learned about glycolysis, we discussed the fact that red blood cells rely exclusively on glycolysis for energy production, and that's because they have no mitochondria. And up at the far end of the spectrum, we can see that eggs, and these are eggs that are used from a reproductive sense in humans, can contain upwards of 200,000 mitochondria. And this also is important to highlight that all of the mitochondria that you have within your own body are likely inherited from your mother. Very rare exceptions. A sperm contain only a few tens of mitochondria, and most of them are actually tagged for destruction once they sort of get inside the egg. And so it's very rare that paternal mitochondria kind of persist within a growing organism. So a little bit more on details with mitochondria. If we, again, compare an early prokaryote which did not have organelles to a modern day eukaryote, we can see a lot of homology between maybe the cell membrane and the cell wall that we found in early prokaryotes, so having sort of still 2 membranes here, and then the membranes that we find within essentially what equates to a prokaryotic bacteria that is found within our own cells. And so kind of expanding on these red rectangles here, we can see that we have the same sort of bilayers, two separate bilayers that we have here, reminding ourselves that even though we've drawn a little line here, that this represents a lipid bilayer. But we've got two separate lipid bilayers here, whether they represent the cell membrane and the cell wall inside of a bacteria, or they represent the inner and outer mitochondrial membranes in a mitochondria. One of your homework questions, again, was to kind of organize and have yourselves think about sort of where these different regions are. So just to highlight, we won't spend too much time talking about prokaryotes, but prokaryotes do this same biochemistry. And so they basically have the inside of their cell that's separated by a cell membrane, with the periplasmic space being the space in between the cell wall and the membrane, and then the outside of the cell. So again, they're going to be doing proton pumping, essentially from the inside of the cell to the outside of the cell. And that's going to be different when we talk about mitochondria. Mitochondria, again, the very inside of the mitochondria here is the matrix. Then we have this inner mitochondrial membrane, which is very folded, and that allows us to increase surface area. We've got the outer mitochondrial membrane, which is very porous. And then the space that's in between them is termed the inner membrane space. So we'll use these abbreviations to make things simpler here. IMM for inner mitochondrial membrane, OMM for outer mitochondrial membrane. Inner membrane space is, again, the space that separates the cytosol from the matrix. So just some sort of anatomy of the mitochondria, as it's important to orient with what's going on. So we spent a good bit of time talking about the different players that are involved in the electron transport chain, not necessarily the protein components that are actually doing the electron transport, but the cellular metabolites that are participating. So again, NADH and FADH2 are important because they supply the electrons to the electron transport chain. And then the importance of synthesizing ATP from ADP and PI. So we wanted to talk about where these pieces come from, where they need to go, how that presents an issue if it does, and what the solution is. So again, talking about NADH, a little bit is produced in the cytosol, again, from glycolysis. The bulk is produced already in the mitochondria by pyruvate dehydrogenase, citric acid cycle chemistry, and then beta oxidation, which we'll learn about in a couple of learning units. So for the most part, these guys are made where they need to be. But for any NADH that's produced cytosolically, that's problematic because it gets delivered to complex 1 in the electron transport chain on the matrix side, so all the way on the inside of the mitochondria. So that's problematic because it's in the wrong spot. What's important to highlight, for many of these very important species, we're going to see that you can't actually move that species directly across a lipid bilayer. You might have to disguise it as something else, or there's a very specific channel that allows it to be transported. In the case of NADH, we're going to use the malate shuttle, and we've kind of equated this to thinking about it being like a drug mule. We actually don't have NADH pass from the cytosol into the mitochondria or into the matrix space of the mitochondria. What we're actually going to have happen is it's going to shuttle its electrons, those electrons sort of being like the drug that we're going to have, and then malate being sort of the mule that carries it. So we'll see that chemistry in a couple of slides here. FADH2 we learned about, interestingly, in our last chapter, because that again is really delivering electrons directly to Q from complex 2. So we talked about FADH2 being generated in the citric acid cycle directly by succinate dehydrogenase, which we actually said was complex 2. So the electrons from FADH2 are actually originated within the electron transport chain. And again, that's going to live in this intermembrane space, really actually in the inner mitochondrial membrane. Those electrons get delivered to Q, which is a mobile electron carrier. So there's actually really not anything to think about in an issue or a complication that we have to have a solution for with the electrons that come from FADH2. Now, thinking about the ATP, ADP, and PI situation. So remember, the whole purpose of oxidative phosphorylation, again, our focus here is in oxidative phosphorylation. So again, we're going to have ADP in the matrix, and then ATP having to go out to the cytosol. So again, if we think about where oxidative phosphorylation is going to occur, that is going to occur by making ATP in the matrix. Again, if we've got substrate level phosphorylation, we've actually already got ATP in the cytosol. But again, if we've got oxidative phosphorylation that's making ATP, it's going to be made in the matrix. So this is problematic if you need to have ATP be used out in the cytosol. So again, we've got to have a way to have ATP get from where it's made in the matrix out to the cytosol, which is where it might need to be used. And so we have an ATP translocator, and we sort of called this our revolving door. So we're going to have a protein translocator that will allow ATP to go out, and as we'll see, ADP to go in. So again, ADP, again, its origination depends on its use. If it's ADP that's generated from ATP hydrolysis in the cytosol, then we're going to have ADP out in the cytosol. Again, but it's more problematic if we've got ADP that we need to get from the cytosol into the mitochondria, less problematic if we've got mitochondrial ADP already. Again, it needs to be in the mitochondria, so that's where it's going to be assembled with inorganic phosphate to make ATP. So again, the issue here is that it depends on where it's made, but again, we need to be able to get ATP or ADP, into the matrix. So again, we're going to use this same revolving door for that. So at the end of the day, remember, ATP is going to be made in the matrix, but we're going to need to export it out. So ADP is often generated in the cytosol from the hydrolysis of ATP. So we need to get that into the matrix so it can be remade into ATP. And because it's made by phosphorylating ADP with inorganic phosphate, this inorganic phosphate, again, needs to get into the matrix. So inorganic phosphate is found all over the place. Again, substrate level phosphorylation, we're going to have it being used in the cytosol. But if we're talking about oxidative phosphorylation, we're going to need to get this inorganic phosphate into the matrix because that's where it's going to need to be used. So again, we need to have inorganic phosphate into the matrix so that it can be combined with ADP to make ATP. So how we get it in there is with this inorganic phosphate proton symport, and we kind of referred this as a piggyback. So remembering that we're normally going to be taking and moving protons from the matrix out to the cytosol, they are going to be wanting to come back in to kind of get rid of that concentration gradient or flow with their concentration gradient. And so if we can have some inorganic phosphate piggyback on a way back in there, that's a way to use that energy from the proton gradient to pull inorganic phosphate in. So that's what we see on this last slide here. So this last slide here just kind of highlights these three different solutions. Our malate shuttle as our drug mule, our ATP translocator as our revolving door, and then our inorganic phosphate proton symport as our mechanism to piggyback and get inorganic phosphate in. So let's just kind of take these one at a time and highlight what's going on. So remember, this was the malate aspartate shuttle. We're not going to be focusing on the aspartate kind of piece of it right now. We're going to be associating malate dehydrogenase, which we learned in our last learning unit on citric acid cycle, has both mitochondrial and cytosolic forms. But again, if we start right here with cytosolic oxaloacetate. So again, if we think here we have actually cytosolic oxaloacetate. So if we start here with the cytosolic oxaloacetate, we can use malate dehydrogenase to convert that into malate, that's cytosolic, move this in via the shuttle here that's in this inner mitochondrial membrane. And then we're going to have malate that's within the mitochondria. We can use the mitochondrial isoform of malate dehydrogenase to convert it back into oxaloacetate. So the important part that we have right here is we started with an NADH here, and we're actually going to be looking at these arrows actually going in this opposite direction. This picture is from our chapter on the citric acid cycle. So we were actually talking about how we got oxaloacetate sort of out here. Actually, I think this was from our chapter on gluconeogenesis, talking about how we get oxaloacetate out into the cytosol for use in gluconeogenesis. So we're actually looking at the same shuttle system, but operating in the opposite direction. So what we're going to see here is we're going to start with NADH that's in the cytosol. Oxaloacetate is really going to be our mule, if you will. The electrons from NADH are going to be delivered to oxaloacetate, reducing it to malate. So again, if the electrons are our drug, they're delivered to our mule oxaloacetate to generate malate. Malate does have a transporter. And once malate's inside the mitochondrion, we can have those electrons, those drugs, be delivered back to NAD+, regenerating NADH. And so the net equivalent is NADH that's produced in the mitochondria, but it is not the same NADH that started on the cytosolic side. So again, oxaloacetate, we could say, is the drug mule for the electrons that are found in NADH. So that's how we handle getting NADH inside. Talking about a revolving door, we can see that we've got this ATP-ADP translocator. So again, we're going to have ADP that's in the mitochondria and ATP that's in the matrix. So again, ATP is being produced in the matrix, but it needs to get out to the cytosol. ADP is often produced in the cytosol from ATP hydrolysis, and it needs to get back into the mitochondrion. So we have essentially this revolving door that we can have these each move in accordance with their concentration gradient. So they will go to where their concentration is lower. But one thing that's very important to highlight here is the net difference in charge, because when we're going to have a TP kind of going out, that's at minimum a -3, if not a -4 charge, and it's a difference of a -1 charge with ADP. So the net, if we're looking at this revolving door being a 1-to-1 system, is that the net is moving a charge of -1 out. And this is going to become important later as we talk about sort of this chemiosmotic theory. So the idea that we're not only pumping a concentration gradient of protons, but we're also dealing with the buildup of charge. So the last little piece to talk about here is this piggybacking system that we have with protons and inorganic phosphate. So again, this is the matrix side here, the intermembrane space. And normally what happens is we are pumping protons via the electron transport chain from the matrix out to the intermembrane space. So there's normally a high concentration of protons out here, meaning a low pH. Some of those protons can be used in this proton inorganic phosphate symport to allow inorganic phosphate to be moved against its concentration gradient and built up in the matrix space. So again, when we talk about symports, it's usually moving something down its concentration gradient to allow something else to be moved up its concentration gradient. So symport meaning they're moved in the same direction, but in this way too, we could say that this is secondary active transport that we have for this inorganic phosphate, because we are using the gradient from another molecule to be able to pull something in. So again, we're going to have the protons being returned to the matrix. That is sort of downhill. Inorganic phosphate being moved into the matrix is uphill. Again, we can think about this as being facilitated diffusion for the protons, but we're actually going to be doing secondary active transport to move these inorganic phosphates against their concentration gradient. So we wrapped up this initial sort of lecture period just doing an overview of electron transport. So an important equation to kind of remember is the net of what happens in the electron transport chain. And remember, we liked putting it in this order because that allowed us to think about the different pieces and then what's happening. So again, electrons come from NADH and FADH2. They each lose their electrons, so become NAD plus and FAD. The purpose of moving those electrons through the electron transport chain is to translocate protons from the matrix into the intermembrane space. And then we also see that oxygen is going to be the final electron acceptor, and it's going to be reduced to water. So an easy way to remember that overall equation that we have for the electron transport chain. Kind of looking at, and another homework question that you had and something that you need to know is thinking about kind of drawing a pathway out for what happens. So again, electrons starting at NADH, they're delivered to complex 1. Complex 1 is capable of generating a proton gradient. We talked about how that occurs, and we'll get to the details of that in just a little bit. But conformational changes are responsible for moving protons in complex 1 from the matrix to the intermembrane space. Electrons then are delivered to coenzyme Q. Complex 2 also delivers electrons to coenzyme Q, and from this point, it's the same sort of process here. Electrons from Q go to complex 3, and this delivery of electrons from Q to complex 3 creates something that we call the Q cycle. We didn't get into the details of how the Q cycle works, but at the end of the day, the Q cycle is what's responsible for generating a proton gradient and translocating protons for complex 3. You'll note here that complex 2 does not pump any protons, and so electrons that come in via complex 2, missing that first complex 1, are not worth as much. So as a heads up, we'll get to this later, NADHs are worth about 2.5 ATPs in terms of the amount of protons that they pump. FADH2s are only worth about 1.5. Electrons from complex 3 are delivered to the mobile carrier cytochrome C. This again is a mobile carrier that lives in the inner membrane space. Those electrons then go to complex 4. We should have highlighted here that we do have a proton gradient that is synthesized via complex 4. So highlight here that there is a proton gradient that is synthesized there. This is what we call vectorial protons, so protons that are actually physically moved. But we also have protons that are contributing to the gradient by being removed from the matrix as those electrons are delivered to oxygen to generate molecular water. So again, an important equation that you need to know in terms of the flow of electrons through electron transport. Again, we can see here with a little bit more detail as we map this on to sort of a map, if you will, of where everything is. Again, we can see that NADH enters its electrons from the matrix side. That's why it was so important that we got those electrons via our malate aspartate shuttle to be as NADH on the matrix side. Again, whether it's from FADH2, again being succinate dehydrogenase from the citric acid cycle, or from complex 1, we have our electrons end up at coenzyme Q. Again, coenzyme Q being a mobile carrier, but a very hydrophobic mobile carrier because it lives in this inner mitochondrial membrane within this lipid bilayer. Deliver them to complex 3. Again, we're going to have the Q cycle that we have here. Again, those electrons delivered to our second mobile carrier, which is cytochrome C, very hydrophilic protein, so it lives within the inner membrane space, delivering those electrons to complex 4, generating a proton gradient here by moving protons that we call vectorial protons, and also using protons in chemistry, we call those scalar protons. At the end of the day, we've increased the concentration of protons in the inner membrane space, decrease the concentration of proteins in the matrix, thus making the pH in the matrix much higher. We're going to move on to next to talk about what actually happens in the oxidative phosphorylation process. But what really happens there is we release those protons back through the inner mitochondrial membrane into the matrix. They're going with their concentration gradient. And we're going to use that energy to run this molecular machine and synthesize ATP. So again, moving on and thinking about some energetics that we have here with electron transport. We spend a little bit of time talking about some electrochemistry, or what you might remember as electrochemistry from maybe your general chemistry course. And if we think about what happens here, we've got two standard reduction potentials. We have the standard reduction potential for NAD+, and remember, a standard reduction potential has to do with the energetics of a certain species taking on electrons. And so these are both written as standard reduction potentials for NAD+ taking on electrons and oxygen taking on electrons. We can see that it's actually more energetically favorable for NADH or NAD+ not to take on electrons, but to give them up. And so we see a reversal of this redox reaction, to write it not as a reduction, but as an oxidation. And so we flip its sign. And when we sum these up, we get the net overall of what happens from a redox standpoint, from the starting point of electrons being the oxidation of NADH to the ending point of electrons being the reduction of molecular oxygen. And we can see that we have a very favorable, meaning positive, standard reduction potential. So remember, delta G's are favorable if they're negative. Delta E's are favorable if they're positive. But we have a nice equation here that allows us to relate delta G's to E's. So this is delta G equals minus NFE. And if we actually do this chemistry, taking into account the number of electrons that are transported and then the Faraday's constant that we have here, we can see that each NADH is worth about 218 kilojoules per mole. If we think about the fact that, one NADH, even if it was worth 3 ATPs, would be 91.5 kilojoules per mole, which is significantly less than the amount of energy that we potentially have. But remember, these are not 100% efficient processes. We're going to lose some of this energy as heat. So now moving on and talking about a little bit of detail with each of the electron transport chain complexes. So we're going to be taking this entire figure here, specifically just for electron transport, and I've highlighted some of this already, but we'll just kind of go through it quickly to talk about each of these complexes in turn as electrons are passed through them. So again, complex 1 that we have here, we've got this first complex that we have here. We've got electrons coming from NADH on the matrix side. They're going to go through complex 1 and they're going to be delivered to Q. What happens in this protein complex to allow us to translocate protons is we have redox changes. Redox changes cause conformational changes. Conformational changes shift pKa's, and then when we shift pKa's, that allows us to either take up or release protons. So again, we're going to take up protons from the matrix, and then we're going to ultimately deliver them to the inner membrane space. Complex 2, and again, we can see complex 2 here is really succinate dehydrogenase from the citric acid cycle. No proton pumping that happens here. We just have these electrons ultimately get delivered to Q, which is our hydrophobic mobile carrier. So again, coenzyme Q here, sometimes called Q, sometimes coenzyme Q. Sometimes we'll see it referred to as QH2 if it's in its reduced form. It gets its electrons, again, either from complex 1 or complex 2, but ultimately it's going to deliver its electrons to complex 3. At this point, we kind of have that there's no mechanism of translocation available. What we'll really see is complex 3 with Q. These two together kind of allow us to translocate protons in what we call the Q cycle. So we've got kind of a unique movement of electrons here with these blue arrows. We didn't learn any of the details of this really neat system, but it allows us to increase the thermodynamic efficiency of this process by pumping twice as many protons as we might normally be able to do. And so again, we're going to have Q deliver its electrons to complex 3, go through this Q cycle. We're going to pump twice as many protons as we could, and ultimately deliver those electrons to our mobile carrier cytochrome C, which is a very hydrophilic carrier, and so it lives in the inner membrane space. So again, cytochrome C here as a mobile carrier takes electrons from complex 3, delivers them to complex 4, so there's no proton translocation that happens here. And then finally in complex 4, we're going to have electrons that come in from cytochrome C, and they're going to ultimately be delivered to molecular oxygen. What's kind of unique about this complex, and we've already highlighted it on a few other slides, is we do have the movement of protons, and we call those vectorial or pumped protons. So we actually have a physical translocation of some protons to contribute to the proton gradient. But we also have some protons that are actually taken up on the matrix side to help contribute to the proton gradient by just removing them from the matrix, not moving them over to the inner membrane space, but just by eliminating them from the matrix side. We call these scalar or chemical protons, and again, they're used in combination with molecular oxygen, and we should probably say plus electrons here as well, to generate water. So busy slide, I know here, so let me sort of break it apart here in terms of the content that we talked about. This is a very simple representation of the electron transport and oxidative phosphorylation processes. So again, electron transport here, we've got highlighted in red, and they really only have the complexes that are responsible for generating a proton gradient. So that's 1, 3, and 4. Oxidative phosphorylation here is what's shown in blue. So we've got complex 5. We haven't really talked about the details of that. We've talked very generally that we are going to use these protons that are going to come back through this complex with their concentration gradient, and we're going to use that energy to synthesize ATP. So again, that's oxidative phosphorylation. So the idea of these two processes being coupled is very important. And so a couple of the things that we're going to highlight here, again, just a few of the other things that are on the slide here. Remember, electron transport, the purpose is to make a proton gradient. Oxidative phosphorylation, the purpose is that we use this proton gradient to run an ATP hydrolysis machine in reverse, and that's how we're going to make ATP. A couple of other things to highlight. We looked at this scary equation, but we said, remember, don't be scared by it, okay? Delta G really is a reflection of the difference in the concentration between protons. So again, we've got protons that are going to be higher in our outside compartment here than they are in our inside compartment. And so this creates a concentration gradient. So again, this piece really represents the thermodynamic driving force by our concentration gradient. And this other piece is really important here, and that represents the fact that we are not only pumping chemical species into a concentration gradient, but those species that are being pumped are charged, and so we're going to build up a charge gradient. And so why this is very important is it allows for sort of an extra thermodynamic driving force in the fact that we can build up not only a concentration gradient, but a charge gradient across this inner mitochondrial membrane. So what this means is we can have a delta pH, a change in the concentration of proteins or protons that's only about 0.75 pH units, and that's sufficient to effectively drive the synthesis of ATP. So again, looking a little bit more detail on this equation that we have here. Again, delta G equals minus RT ln of KEQ. So again, this equilibrium constant, really the process that we're looking at is the difference in the concentration of protons that are on the inner membrane space versus the matrix. That's how we define the proton gradient. And again, that proton gradient is generated by electron transport moving or allowing for the movement of protons from the matrix into the inner membrane space. So as we highlighted here, the fact that these two processes, electron transport to make the proton gradient and oxidative phosphorylation to use the gradient to synthesize ATP, really important that these are coupled. And I know concepts that we've talked about and we've seen kind of threaded within different learning units that we've had in both Biochem 1 and Biochem 2. We see this process of uncoupling, or disconnecting a proton gradient that's generated by electron transport and used by oxidative phosphorylation. So again, uncoupling proteins are a very natural mechanism that we know exists for that. And then we talked about 2,4-dinitrophenol as a synthetic drug that was able to do the same thing. Both of these provide alternative channels for protons to get back into the matrix from the inner membrane space. All right, then we moved into a little bit more of an understanding of how this complex 5 or this ATP synthase or this ATP hydrolysis machine that we're running in reverse works. So again, highlighting what we really have here, coming back here, that's this guy. Okay, so I wanted to highlight and move back to here because we're going to be looking at this guy really upside down. So the pictures that I'm going to have on the next few slides that we're going to discuss are going to show this guy with his lollipop head in the air. But I always want you to remember that that lollipop head, that lives in the matrix. That's the innermost part of the mitochondria. Okay, but unfortunately when we have this little cartoon, I didn't want to put it upside down because it was then confusing to read the words, but this represents really the inside or the matrix part. Okay, we call this domain, this lollipop head, this F1 domain, and then we've got this F0 domain that lives in the inner membrane space. And again, it's a little bit confusing here because this is an animation and a cartoon that they have from a bacteria. They have cytoplasm and then this periplasmic space. So again, cytoplasm is really all the way on the inside. Let me go back just a minute to, I know I'm flipping very quickly, but I want to make sure to highlight this. Okay, they're looking at this in a bacteria. And so the inside really is the cytoplasm of the cell. And then there's that periplasmic space. So let me go back then to where we were here. So just to highlight that this cytoplasm really equates to the matrix inside of a mitochondria. And then this periplasmic space really equates to the inner membrane space. Okay, so a little bit of detail on these two domains. This F1 domain, we call it the lollipop head. We basically have these Greek letters that refer to these orange segments, if you will, alpha and beta segments that we have here. There's going to be 3 sets of them. And these proteins actually toggle between a loose, tight, and open conformation. Loose, tight, open, loose, tight, open, loose, tight, open. And so the loose conformation is capable of binding ADP and PI. The tight conformation you can think about as squeezing them together to synthesize ATP. And then the open conformation allows them to be released. And that's what we kind of see here in this temporal sequence. Imagine that you're looking down on the top of this guy. We're kind of rotating this so that we can look down on the top of it. That's what we see here. So this little delta hat here really represents this green guy here. And these each represent an alpha beta set of orange wedges that we're looking down upon. And what we can see is that we toggle between a loose to tight, and then tight goes to open. So again, we've got a temporal sequence that goes loose, tight, open, loose, tight, open. Now, we talked about this in class, and it's a little bit counterintuitive. You might think that the loose to tight, that squeezing sort of chemistry, might be the one that costs money, but it's actually not. Going from the tight to open, sort of product release, is actually the step that requires energy. And so we're going to see that that's the step that requires really the energetics of movement of protons. So again, this lollipop head is where ATP synthesis occurs, but translocation of protons occurs in this F0 domain. And again, to help you remember, we got a little 0 there, reminds us that this is where we've got this merry-go-round that really is going to rotate here. So we've got this merry-go-round, and you can think of each of these little C domains representing seats that we can have protons go and sit on. So again, these letters, we're going to see not Greek letters, but regular letters that we have here for the different domains. We've got A, we've got B, and then the C domains are really the important domains. Those are the seats in our merry-go-rounds. And we're basically going to be moving protons from the inner membrane space, returning them to the matrix. And we're going to see in a slide or two here how that happens. We're going to talk a little bit about the details and some specific amino acid residues that facilitate that process. But at the end of the day, just like a merry-go-round, this operates in a circular motion. This rotation that we have here is imparted energetically to the conformational changes that are needed to occur in the F1 domain to allow us to translocate between this loose, tight, and open conformation. So again, a little bit of details here, a little bit in the weeds about how we can have proton translocation cause this molecular motion and this rotation of this circular sort of set of proteins. So again, we've got an aspartic acid residue that we're going to have here. And so again, thinking about aspartic acid, it's normally negatively charged. And so when we bind protons here and we protonate this aspartic acid residue, that changes from a negative to a neutral, and that induces a conformational change in that subunit. So remember, we talked about a lot of this. Changing protonation state on proteins can induce conformational changes, and we see an example of it again here. So we can think about that sort of being a push, if you will. We have sort of a bending in this chair, if you will, as a proton sits down, and that pushes it forward. Just like if you're thinking about a merry-go-round, right? You can make a merry-go-round move by either pushing on the bars or pulling on the bars. So here's the push. Again, we see a bulge that happens when we have that conformational change. It pushes against an adjacent subunit, and then that rotation is pushing that steric hindrance away. So we're going to have one trip around by a proton that's sitting in a C subunit. When it gets to the other side, it exits the ride. What happens there is now we're going to have that aspartic acid residue go back from being neutral to negatively charged. There's an arginine residue here. Remember, arginine residues are positively charged. So now when we have that negative aspartic acid residue getting near that positively charged arginine, now we have our pull. And so this is really a pull that we have here to finish pulling this empty seat. Now it's ready to bind the next proton. So kind of a really neat application of protonation-induced conformational changes in proteins. And then remember, we watched that really fun movie. We kind of talked about taking one of these amazing molecular machines, using a His tag to fix it to a glass slide, using a cysteine and a biotin to attach to streptavidin, which was covalently linked to a very long fluorescent actin filament. You feed this machine ATP, and you're going to whip this actin filament around in a circle. And we were actually able to watch that using some some microscopy, fluorescence microscopy. So a really neat little experiment that we were able to kind of watch a movie to kind of see in action. So we've talked about this a little bit already, so I'm not going to say too much more about it, but this is just highlighting the two different mechanisms that we've talked about for uncoupling, reminding us again about this drug, 2,4-dinitrophenol. 2,4-dinitrophenol, in becoming protonated, is allowed to more easily diffuse through the lipid bilayer, and again can return protons in that case to the matrix. We also can see here's the normal kind of translocation of protons through the electron transport chain. And if we have not protons coming back through the ATP synthase machine, but rather coming back through an uncoupling protein, we can have dissipation and uncoupling of electron transport's proton gradient generation and the utilization of that proton gradient to synthesize ATP. So again, but this dissipation of a proton gradient can't be without that energy doing something. So again, this protein is called thermogenin because its purpose is to generate heat. So the last thing that we sort of talked about is this concept of what we call an OP ratio, and that's the ratio between the oxygen used, which is a reflection of how much electron transport is going on, and then ATP produced, which is a reflection of how much oxidative phosphorylation is going on. Again, these two processes are coupled or connected by the proton gradient. And so as you see a change in the OP ratio, that means this proton gradient is not exclusively coupled between these two processes, but you might have other things going on, maybe like this uncoupling protein or the use of a drug that translocates protons back across that inner mitochondrial membrane. So what we have here in our last little section here is a discussion of how we are going to regulate electron transport. Now there's lots of different mechanisms we could talk about. There's lots of different aspects of regulating electron transport. I'm just going to focus on one here just for simplicity's sake, and that's the discussion of using an inhibitor to the F1 domain. So again, remember the F1 domain is the area in ATP synthase, complex 5, that's actually going to be synthesizing ATP. So there's a molecule that exists within the matrix that's able to inhibit this domain. So if this inhibitory domain is as a tetramer, it's inactive. Now what happens is if it encounters a high concentration of protein or protons, then it will dissociate into an active dimer. So where this can get confusing is, remember, this is a molecule or a substance that's intended to inhibit oxidative phosphorylation. So if we have a lot of protons that are in the matrix, right, that must mean that there's not a lot of electron transport happening. So again, a drop in the matrix pH, low pH, means there's a high concentration of protons there, which means there's not a lot of electron transport happening. If there's not a lot of electron transport happening, we want to make sure that ATP synthase is not trying to synthesize ATP, or we should say is not really active. Because if this machine is active, it could actually run in reverse. It actually could run in the way that it's intended to operate, which is to hydrolyze ATP. And this would be a futile cycle. So this one can be a little bit confusing to think about. So remember, we're talking about this inhibitor is active at low pH. Again, low pH means not a lot of electron transport happening. If we don't have electron transport happening, we don't want this oxidative phosphorylation machine to be operational, because it could operate in a direction that's going to allow it to hydrolyze ATP. And again, if we're in a nice resting state, not doing a lot of electron transport, we want to make sure that the ATP synthase machine is quiet. We also talked very generally in this last little section here about molecules that might inhibit complex 1. We'll spend some more time actually talking about these much more towards the end of the course. We'll talk a little bit more about metformin over the next couple of chapters. Metformin is a drug that's given very often for patients who have type 2 diabetes. And so we'll talk about all the different places that metformin seems to have kind of an effect. And one of the places that it does have an effect is by inhibiting complex 1. Now again, when we say inhibiting complex 1, you don't want to completely inhibit complex 1. That would actually be fatal, and that's what things like cyanide do, or rotenone for that matter. But metformin is one that just inhibits complex 1 enough to allow us to see some interesting biochemical processes happen. And so we'll spend some time over the rest of the course kind of digging into the details of those. So one of the things that it was important to think about is when you start doing chemistry where you have a lot of oxygen around and a lot of electrons kind of flying around, there's a potential to make some kind of reactive oxygen species that could be problematic. So we defined the term obligate aerobe, and we are obligate aerobes. We cannot exist without oxygen. We can kind of operate for short bursts of time without oxygen. We can sort of operate in an anaerobic sort of state for short periods of time, but not for long periods of time. And so when we kind of talk about the costs of aerobic metabolism, we can think about that in sort of two ways here that we're going to discuss. Lots of applications here, but two that we're going to focus on. One is more sort of the physiologic aspect of impeding blood flow can cause cell death. And it can cause cell death within minutes because there's certain tissues, especially the heart and the brain, that cannot operate anaerobically. And so if you deprive those tissues of oxygen, it can be very problematic for those two tissues. The other one that we're going to spend a little bit more time talking about is the generation of reactive oxygen species. And again, this is an unavoidable byproduct of using oxygen. So three that we're going to talk about here, and they're kind of ranked here in terms of their ability to do sort of oxidative damage. The first is superoxide radical. So again, if you've got molecular oxygen around and you've got electrons around, you have the ability of generating this superoxide radical, which is super damaging. Now, that's problematic, but you actually have superoxide dismutase. That's an enzyme that will take superoxide radical and convert it into peroxide. Well, that's problematic because peroxide can still be damaging. So you have a third enzyme called catalase, which is responsible for converting hydrogen peroxide into water and back into molecular oxygen. And we've talked a lot about glutathione. Glutathione is another sort of biological antioxidant that can deal with peroxide, itself becoming oxidized and then reducing peroxide to water. One of the interesting things about this enzyme that uses glutathione peroxidase, is it has an absolute requirement of selenium. So one of the reasons why selenium is thought to be so important for mitochondrial function. So one of the things that I just want to put a little kind of seed in your brain, if I will, to think, we're going to talk more about these species later. One of the things that we're starting to realize about mitochondria is it was used to be thought that maybe these species that are generated, the superoxide radical, hydrogen peroxide, they just happen to be reactive byproducts, and thank goodness we've got these mechanisms to get rid of them. But one of the things that we're realizing now is maybe these are important chemical messengers, some signals that are actually coming out of the mitochondria. Remember, these are ancient bacteria, so they have an amazing ability to be able to communicate what's going on, sort of the health of the inside of a mitochondria, by how much or how little of these reactive oxygen species that come out. So it's a really interesting field of research now to think about maybe these reactive oxygen species aren't all bad. Maybe they're actually important chemical communication signals that allow the mitochondria to communicate with the rest of the cell and to affect changes that are necessary for important biological function. So, kind of a neat picture I got from a book that I just started reading on navigating metabolism, is what it's called. But it was a really neat little picture that kind of highlighted the differences between processes that happen within a normal mitochondrion and then one that's sort of dysfunctional. And we've talked about this a little bit. We learned it in the citric acid cycle learning unit, this concept of the Warburg effect. And one of the things that we see with the Warburg effect is that there's an increased production of lactate and a decreased production of or utilization of oxygen. So cancer cells, for example, are well known to exhibit this Warburg effect. They see excessive utilization of glucose, excessive production of lactate, and very little consumption of oxygen. And it originally was thought that, well, that must mean that the mitochondria aren't working. But what we've actually begun to realize is that it's more that the mitochondria are working differently. Certain processes like the citric acid cycle are actually greatly upregulated within cancer cells. And it's not to be used to generate energy, but rather be used to generate a lot of the biosynthetic intermediates that are necessary for rapid cell division. So remember, the normal sort of clockwise direction of the citric acid cycle is used for catabolic processes to break fuels down. Running the citric acid cycle in the opposite direction can be very important for synthesizing precursors for important things like nucleotides to be able to generate and utilize and do cell division, and lipids to be able to generate those important lipid bilayers to make new cells. So really interesting things that seem to highlight that the Warburg effect doesn't necessarily tell us that mitochondria aren't working, they're just working differently. And so again, we spent a little bit more time just kind of tying a lot of these pieces together with our last lecture here. Again, reminding us a few concepts about this endosymbiotic theory. And one of the reasons that provides sort of some evidence for what mitochondria are doing is that this endosymbiotic theory really sort of happened before oxygen was available. So it was less maybe that these mitochondria that were kind of captured by early bacteria were captured because they could do electron transport and oxidative phosphorylation to generate ATP. Maybe it was more the chemistry that was going on was an ability to trade metabolites. So again, less that they're doing this electron transport and oxidative phosphorylation chemistry, but maybe it was more the citric acid cycle chemistry. Maybe it was the more this kind of biosynthetic sort of processes that were of advantage. So again, some important roles for mitochondria that we know of, right? They do have an important role in generating ATP by electron transport and oxidative phosphorylation. So we're not saying that that's not important. That's an important kind of classical biochemical function of mitochondria. But what we're saying is that there's a lot of other things that are going on that maybe we're not giving enough attention to. And one of them is the fact that mitochondria can be this important biosynthetic hub, right? The chemistry that can happen with a lot of the biochemical processes that happen in mitochondria can allow us to synthesize glucose, lipids, nucleotides, and amino acids, basically all of the building blocks that we need to make new cells. So if you want to undergo cell growth and cell division, you need to make these. So maybe these were the important metabolites that were important to kind of take advantage of in a symbiotic relationship. Again, maybe mitochondria are really important in using some of these metabolites to, talk about the overall health of a cell, the health of that mitochondria. Remember, we're building up charge gradients in that inner mitochondrial membrane. So that membrane potential is a way of kind of dictating what the health is of the cell. And one of the things that we know is as that membrane potential falls, mitochondria become tagged with ubiquitin. We'll learn about ubiquitin in a couple of chapters here, a couple of learning units, but ubiquitin is a tag that is used to attach onto proteins that need to be tagged for destruction. So anything that gets slapped with the ubiquitin tag is tagged for destruction by the cell. And so if mitochondria are not doing very well, then they get tagged with ubiquitin and your body can recycle them. And speaking a little bit more again about these reactive oxygen species, there's a lot of research out there suggesting that these are important signaling molecules. So again, we talked about reactive oxygen species being important signaling molecules, but a couple of other things that I want to highlight that we really didn't spend too much time talking about, and maybe we'll spend some more time at one of our advanced topics at the end of the class, is talking about 3 other things that mitochondria can kind of spit out as a way of using in a signaling pathway. So for example, mitochondrial DNA. If mitochondrial DNA is released from mitochondria, that elicits a massive immune response. And maybe that's an important process in signaling for cellular apoptosis. Same thing with releasing cytochrome C. Remember, that's that hydrophilic mobile carrier for electrons in the electron transport chain. However, if it's released from mitochondria, this is a cellular signal for apoptosis. So again, we can have a lot of pieces that normally exist inside of a mitochondria. If released, they can signal for important events like eliciting an immune response or signaling for apoptosis. Calcium is again another one of these things that if it is released from mitochondria is an important cellular signal. So highlighting again what we kind of showed on that first slide, but just kind of written in our own words here, kind of thinking about what happens in normal functioning mitochondria. In normal functioning mitochondria, we've got glucose that undergoes glycolysis to generate pyruvate. Pyruvate will undergo oxidative decarboxylation by pyruvate dehydrogenase to generate acetyl-CoA. Sort of a clockwise motion through the citric acid cycle will decarboxylate these fuels, generating NADH and FADH2, go to the electron transport chain. Electrons ultimately delivered to oxygen to generate water, and that proton gradient is used to synthesize ATP by oxidative phosphorylation. So 2 things to highlight here, 2 things that are very important to think about is the importance of regenerating NAD+. On the short term, in an anaerobic environment, we can very easily regenerate NAD+ by using lactate dehydrogenase to shunt pyruvate into lactate. And again, lactate being reduced allows NADH to be oxidized. That can be a very short-term way to regenerate NAD+, but it's not very sustainable. So in normal functioning mitochondria, the electron transport chain is the main pathway for regenerating NAD+. And again, this is a sustainable mechanism for getting more NAD+ to go to glycolysis. But remember, the main point of glycolysis is not necessarily the long-term extensive production of ATP. So if we think about what happens if we impair electron transport, so again, impairing electron transport means that we're going to be impairing the ability of the electron transport chain to regenerate NAD+. We're going to be impairing the ability of electrons to be delivered to molecular oxygen and generate water. So again, if we inhibit electron transport, we'd expect that oxygen consumption would drop, and we'd have to increase the production of lactate because this now becomes the main pathway for regenerating NAD+. Again, if we now don't run the citric acid cycle in a clockwise direction, but run it in reverse, that allows us to generate citrate. And as we'll see in upcoming learning units, generating citrate is an important building block when exported out to the cytosol for synthesizing a lot of important metabolites. Again, pyruvate we know can be synthesized into oxaloacetate by pyruvate carboxylase. This again is a mechanism that allows us to generate lots of glucose. And again, we're going to have some important chemistry here when we have glutamine being deamidated to generate glutamate. Glutamate is the alpha keto acid of alpha ketoglutarate. And so again, we have ways here of being able to generate the amino acid building blocks that we need. So lots of important biosynthetic chemistry can happen within the mitochondria. Lots of anabolic processes that can happen. We've traditionally thought of things happening in a catabolic pathway. But again, if we inhibit the electron transport chain, we can do a lot of chemistry that allows us to do anabolic processes, which again, if you're a cancer cell, is a really important thing to think about. So that wraps up our learning unit on mitochondrial function, electron transport, and oxidative phosphorylation. And importantly, in our very end couple of slides here, we were really thinking about what it really means if we alter mitochondrial function.